Assalamu alaikum and welcome everyone to the second part of Molecular Biology ASCP Preparation Series, Molecular Techniques. I'm Dr. Lai Abdelati and I'm really excited to share my knowledge and experiences with all of you through this series. So, let's dive in together. Today, we'll continue exploring multiple choice questions covering Part B in polymerase chain reaction optimization. This section will focus on fundamental concepts such as the effect of high or low concentration of DNTBs, magnesium chloride, and primers in a PCR reactions, as well as the PCR contamination. All right, let's move on. All right, here we see the first question we have today. Let's see it. How does a low concentration of DNTBs affect the elongation step in PCR? The correct answer is slows down or halts the elongation process. Actually, during the elongation step, DNA polymerase incorporate DNTBs to sensitize the new DNA strand, right? Okay. If DNTBs are in low concentration, that means the polymerase may not have enough substrate to continue elongation efficiently. This leads to slowing down or stopping the process. All right, let's move on. All right, all right, let's see the next question here. What is the potential consequence of having too low concentration of DNTBs in a PCR reaction? The correct answer, decreased yield of PCR product. Actually, DNTBs are the building blocks of new DNA strands, right? So, insufficient DNTBs can limit the amount of DNA sensitized, resulting in a lower yield of the PCR product. Okay, let's move on. Okay, the next question here. What may be effect of low DNTBs concentration on the overall viability of PCR? The correct answer is decreased fidelity due to incomplete sensitivities. So, low DNTBs concentration can lead to incomplete or incorrect DNA synthesis, okay? Reducing the validity of the PCR amplification as the polymerase may incorporate incorrect nucleotides or stall during the synthesis, okay? Let's move on. Okay, let's see the next question here. What is the effect of using too high concentration of magnesium chloride in PCR? The correct answer is increased non-specific amplification. As we know, excessive magnesium chloride can stabilize non-specific binding of primers, leading to increased non-specific amplification. Okay? Okay, the next question here. How does overlay concentrated magnesium chloride solution affect the annealing step in PCR? The correct answer, it increases primer dimer formation. All right. As we know, high level of magnesium chloride can lead to increased primer dimer formation during the annealing step because it stabilizes non-specific interaction between primers. Okay, which can combat with the specific primer template binding and reduce the efficiency of the reaction. All right. Let's move on. All right, next question here. What is the potential consequence of using too low a concentration of magnesium chloride in PCR? The correct answer is reduced enzyme activity. Magnesium ions are essential cofactor for the DNA polymerase enzymes, as we know, right? So, without sufficient magnesium chloride, the enzyme's activity is hindered, leading to reduced efficiency in the DNA synthesis. Okay? All right, next question here. How does low magnesium chloride concentration affect primer annealing in PCR? The correct answer, it prevents primers from binding to DNA. Actually, adequate magnesium chloride levels are necessary to stabilize the interaction between primer and DNA template. So, low magnesium chloride can lead to poor primer binding. Okay? Okay, next question. What is the potential consequence of having too high a concentration of primers in a PCR reaction? The correct answer, enhance formation of non-specific product. Actually, high concentration of primers can lead to the formation of non-specific product. All right? This occurs because the excess primers can allele to non-target sequences, okay? Resulting in the amplification of unintended DNA fragments. Okay, let's move on. All right. Which of the following issues is commonly associated with the excessive primer concentration in a PCR reaction? Actually, excessive primer concentration can lead to formation of primer dimers. All right? These are artifacts formed when primer anneal to each other instead of the target DNA, which can compete with the desired amplification and reduce the overall efficiency of the PCR reaction. Okay? Let's move on. If you found this content helpful, don't forget to like, share, and subscribe to our channel. Your support means the world to me and help me create more valuable content for you. Thank you for being part of this journey. Let's continue. Okay, next question here. What is the potential consequence of having too low a concentration of primer in a PCR reaction? All right. Low primer concentration can lead to reduced yield of the PCR product, right? Because there is no enough primers to initiate the amplification process efficiently. All right. Let's move on. Okay, next question here. Which of the following issues is commonly associated with the insufficient primer concentrations in PCR reaction? Actually, insufficient primer concentration can result in incomplete or no amplification of the target sequence. This is because there are not enough primers to bind to the target DNA and initiate the replication process, specifically if the target DNA is present in low quantities. All right, let's move on. Okay, what is the common source of contamination in PCR reactions? The correct answer is aerosolized DNA from previous reaction. Actually, aerosolized DNA from previous reactions can contaminate new reactions, leading to false positive and non-specific amplification. All right, let's move on. Okay, the next question. What is the purpose of including no template control in PCR? The correct answer is to check for contamination. Okay, a no template control contains all PCR components except template DNA. Okay, so if amplification occur in NTC tube, it indicates contamination in the reagent or environments. All right. Okay, next question. Which of the following practices can help reduce the risk of contamination in PCR? The correct answer is preparing master mixes in a separate area. All right, let's move on. Okay, next question here. What is the effect of using UV light in a PCR setup area? The correct answer, it degrades contaminating DNA. All right, let's move on. All right, how can you minimize the risk of contamination when handling PCR reagents? The correct answer, by wearing disposable gloves and changing them frequently. All right, let's move on. Okay, the next question here. Which additive can be used to help with GC-rich templates in PCR? The correct answer is DMSO, okay? DMSO helps to reduce the secondary structures in GC-rich templates, improving the efficiency and specificity of the PCR, okay? Let's move on. Okay, next question. Which of the following substance is not inhibitor of PCR? The correct answer is ethanol, okay? Actually, substances like hemoglobin, heparin, xylene, phenol, cyanol, and bromophenol blue are all known inhibitors of PCR. Okay? All right, that's all for today. I hope you enjoyed this lecture. Please don't forget to like, comment if you have any questions, share, and subscribe to our channel to receive all the latest lecture. I also encourage everyone taking the test to share your question with us so that everyone can benefit. Salam.